Buenas tardes a todas, todos y todes. Es mi
Good
agrado
morning, ladies and gentlemen. It is now our fifth and last day of the National Congress of Biochemistry and Similar Majors. I'm Diego Salas Pruging, and now we have Dr. José Onuchic, biophysicist from Brazilian North American. He's at the University of Sao Paulo and the Technological University of California, Caltech, where he got his PhD. Currently, Dr. José Onuchic acts as a physics professor and Olga K. Weiss and the University of Rice. It's a great honor to have you here with us, and I'd like to personally thank you for your presence. For those who don't know him, José Onuchic is world recognized in protein folding, and he's going to be surprising us with his lecture, which is called Protein Sequencing, Coevolution, Energy Landscape, and also structural maintenance chromosome proteins connection. Don't forget to leave your questions in the chat. And without any further ado, I will now leave the floor to Dr. José Onuchic. Thank you so much. Please, the floor is all yours.
Thank you very much. I'm just start to share my screen. It's a real pleasure to be here today and be able to talk to you. And I thank for the invitation from all the students that for inviting me for this presentation. I want to tell a little bit about coevolution. That's a very interesting, important point, and you're going to see as I talk about it, how that has been involved on protein structure prediction and also understanding protein function and protein dynamics. So first of all, I want to say that I'm located at Rice University, and our center is on the 10th floor of this building. That's the Bioscience Research Collaborative. That's a building that we put interaction between the Rice and the Texas Medical Center. As you can see from here, our building is located here. All the back to Texas Medical Center was 100,000 people work on medical research. So that's the area we interact. We have a great visiting program. So sometimes if you come to this area, if you come to the US, please come visit us in Houston. The problem of protein folding is a problem I have been working for many, many years and starting the past. And the basic idea that you have into protein folding is that when the protein gets organized, you can see there's a correlation between energy and order. That means most of the contacts that are formed towards the native structure, they are attractive contacts. That's what creates the principle of minimal frustration and the idea of the funnel that tells you, if you think about this horizontal x, that's the number of states. We call configurational entropy because the logarithm of the number of states. But as you go down in energy, you reduce the number of states and you become more native-like. And that's the concept we call a funnel, right? That basically you go down in energy and you get more order as we start to come down. Because of that, we know that since most of the interactions that stabilize the native structure are attractive, we can create what we call structural-based models that are models that you have the structure of the fold protein. You can create an energy function that tells you the native interactions are attractive and non-native interactions are repulsive. These are called structural-based models. With these models, you can understand a lot about function, folding, states, allosteric, a lot of these things about proteins. So this landscape gives it. That's also the reason why most of the method of structure prediction works when people try to learn from other proteins to predict the structure, is because you always under this assumption that's actually correct, has been proved experimentally very many times, that basically on average, native interactions, that contacts exist on the native structure are attractive and contacts that don't exist on the native structure are repulsive, okay? Now, so if you want to do these structural-based models, you have a limitation. You have to know the structures. So one of the problems you have is that is that basically, although many structures have been done by X-ray, NMR, and all things like that, you don't have enough structures to actually understand every proteins. And actually get even more interesting if you start to go to proteins that are partially disordered, and that means most of these IDPs are proteins that sometimes only fold when binds. That means alone they don't have a structure. How do you understand possible structural motifs of things that are disordered before they bind, they are ordered after they bind? How do you predict these dimer structures and stuff like that? So one thing we have been doing since 2009 is the idea of coevolution, is that basically you can actually understand some part about structures you don't know. You can predict structures based on sequence information. And as you know, we have enormous amounts of sequence information these days. People are sequence everything. You have much more sequence data than you have structural data. So what's the idea? The idea comes into this slide where I show you that if I look here, that's basically the evolutionary tree. You start to see that you have many more sequence that folding motifs. Here is, for example, a particular folding motif that's based just a helix turn helix motif. As you can see, it appears in evolution in many different cases, okay? On situations where the sequences are very different, but the structure is the same. So again, repeating, you have many more sequence motifs than you have structural motifs. Therefore, you can start to think about it, why different sequence motifs give the same structure? If you come the idea of a coevolution, come the following way. Let's imagine that you have here a particular protein structure, and here between red and red, you have a particular contact on that protein. Now I do a mutation into this green group here. What you see is, if I want to keep this interaction attractive, I have to do a second mutation on the pair group. And basically, so that's stabilized. What you see is, you don't have, as the protein gets stable, you don't have a single mutation, but you have a pair of mutations where both of these pairs can be stable. So what happens, we start to look for the things what we call covariance, things that vary together. And when we come to the sequence information, as you can see down here, I'm looking how pair of residues may be changing, how this pair RE may be changed to another pair, and which pairs repeat and which pairs are more stable. That's the idea of coevolution. So the way we do it, and I think that's just a couple theoretical slides. If you don't follow all the details, don't worry about, but that's just something to give you an idea. How do you get this information? What you do first is what you call multiple sequence alignments. You get every particular sequence that folds into the same structural motif, right? So if you have a protein, and the same protein fold in different ways. Basically, in bacteria, you may have thousands of sequences that fold into the same protein. Now you align them. The first thing you do on this alignment is you start to look at every position of the sequence. Let's say there's whatever this position, position I, for example, and I start to see which residue happens more often. So the first thing what you see is what's the most likely residue for that position. And this probability function that we're going to call here Fi tells you that that's the most likely motif. If every residue exists with the same chance, you'd say that the chance would be 1 in 20 because we have 20 amino acids, but that's not the case. So some residues are more frequent than others. Actually, when we do our alignment here, for technical reasons, we actually don't have only 20. I know there are 20 amino acids, but we have 21. Because you have to add the possibility of insertions and deletions, right? So you may have the possibility of having a blank there. But that's just a minor technicality. The second thing is we start to have a look on residues that vary together. That's exactly what I told you about the concept of covariance. So if you have a look here, you can see that now you have this residue. So for example, look at this part S and P, okay, it can change for NI or it can change for KE, but if you tell what's the probability of this pair exists, you see here I'm looking at 6 sequences, and the pair S and P on position 6 and position 17 happens to 6 1/3 of the time. So what we do, we are going to collect all that statistics. Based on that statistics now, I can find a covariance matrix. And the covariance matrix is exactly this Fij minus the local probabilities that tell that. As you can see, if these residues were completely independent, there was no covariance, they didn't vary together, this Fij will be exactly equal to this product and this matrix would be zero, okay? But that's not the case. They vary together. So the fact that when this Fij is bigger gives you that information. The interesting part is this problem that I showed you here, that basically, that tells you that your function is this way, can be written almost like a problem in physics, a problem of spins, where basically covariance two spins acting together. And they have a Hamiltonian, what they call, that can predict these energies, what the people call the Potts Hamiltonians, where this energy comes from pairwise and these terms come from this one. Now, the problem here now is that you have to invert. You want to find these parameters, these energy functions based on all that information. This inference problem is very complicated. And that's something we have done, but we have shown some students, some postdocs in the group. Basically, you're just seeing them here. Farouk Marcus and Martin Waits. Farouk is now a professor. Farouk is now a professor at University of Texas in Dallas, and Martin Waits, a professor in France. They show that actually, in the, what we call mean field in physics, you can show that this energy is just the inverse of this covariance matrix. That made these calculations much easier and allow us now to actually predict what the probability of this context to be formed. And this quantity is this quantity, what we call DI, that I'm showing here. And this DI, as you remember, if you actually took statistical mechanics or entropy, you have this entropy that's P log P, is just normalized by these terms, tells you how likely a context to be formed. So if you're not mathematically oriented, don't worry about, just think about that you're using this information that you got from the system to predict if pairs of contexts exist. Now, what you see here from here is that these predictions are very good. Here is you're showing on about 131 families, you can see that basically my contexts are very good to be predicted. You start to see that basically my predictions are all correct with a probability over 90% as we come down that comes from this information. What I told you about direct information is a particular protein motif, very simple for the sigma 7 factor. And we start to observe in red are the correct context and green is the bad context. What you see is DI that's much better than what people just call the conventional mutual information, because the direct information remove the contexts that are not interactions via a third residues, just only direct interaction of the context. Then when you go, when you do that inversion, I told you about it. So these techniques become very good to predict the structures. You can see from the sequence information, I can get my top contexts that are here. Okay, with that information in hand, I can now continue because as you can see, I can predict well, but let me show you a little bit more about this data. What you can see here is a contact map of this protein. On the bottom part is for this protein RAS, right? Very involved in cancer. And you see in this sort of shaded gray here is the crystal structure of the protein. I overlay my predictions using my DI, using covariance, okay, DCA. As you can see, my predictions in red are very, very good. That's enough to predict the structure of the protein. The green, that are the predictions I believe I was doing wrong. Here is basically top 200 contexts. You see, even most of them are located around the real areas of the protein. There are few of them that are missing, but you can see with this map of contexts, I can predict structures quite well. I proposed that 2009. Technique is becoming very popular these days. Actually, you can see that if I have a protein here and I know its local structures based on if it's alpha helix or beta sheet, just the local part of it, using just this local information, right? Using our structure, make a structural based Hamiltonian where we make this native context attractive using just the contacts from DI, I can predict these structures with 1.5 angstrom resolution. This is great work done by Farouk Marcus and Joanna Sulkowska. They have been on the group, and you can see they do well. Actually, covariance is the main reason that AlphaFold, everything people are talking about it now, has been so successful. Basically, nowadays, the Google people have been able now to create enormous curate databases for sequence information. That's the reason of the success of going this way. So what you see now is that people use all the information you have, not just the structural information that's available to you, but also all the sequence information that was given before. And now that's how things are very, very successful on predicting those structures. So you can see the power of this method by just using sequence information to get information about your structural case. Now, what I want to do now is actually do something more interesting. I want to get out of structure prediction, because I think that's a great field. There are lots of people working on it. We did some work on it. AlphaFold now is engineering. They use these basic ideas we developed more than 10 years ago. They're applying their methods. They are very successful. But what we got very interested is the following situation. I'm getting for the 131 families I told you about it, and I'm looking all the contacts I predict. What's the distance between the alpha carbons between these two things? So what you observe is that this first peak here is just the direct contacts, and I'm fine. Basically, as you can see, they are the order about less than 5 angstrom. That's a direct contact between residues. You have a second peak here that's coming around 8 angstrom, and these are contacts that are separated by water. There are lots of water separate contacts into proteins. Now, but what your interest is about this tail. These are the contacts that DCA, our method of covariance, predict as being contacts. However, they're not native contacts on the structure. They're very far apart. So the question you ask is the following. Is this wrong predictions, or there is something interesting? I got very curious about that, and I came to the conclusion that basically, if you have this context, a very strong signal coming from evolution, come from this covariance from the sequence, it must have a reason. So these contacts that I'm getting down here that appear to be wrong contacts, they're probably contacts involved in multimer structures or in function. Here's a particular case of this protein NTRC1. If you observe in a single protein, these contacts are very far apart. But actually, this protein forms an aptamer, and you can see the interface contact, they come together. So you can see that it was not a mistake by DCA. It was telling that this interaction was actually very important. Didn't exist on that. So what you see is getting this information from DCA may give me not only the information about the native context, but also may give more context, but these contexts may be involved, like on this case, on multimerization, or they may be involved as being functional contexts. So let me give you a few examples of that. Here is a particular case of D-ribose. That's an enzyme that people have been studying for some times. And as people work with this enzyme, they observed a few things here. First of all, I want to point out that basically this top triangle here. This top triangle here means these are the crystal structure of the enzyme. In blue is when this D-ribose is open. The red contacts here are the additional contacts when the enzyme closes. Okay? These are the two structural people exist. They open and close the structure. When we compute our DCA contacts, you observe on this map down here, you observe that the green contact is pretty much the open enzyme. The red boxes that I have here are the contacts of the closed structure. But we have this additional set of contacts here that appears on this green box down here. And the question is, what are these contacts for? So what we did is we did a structural-based model now, where you don't use the structure, but use just the context from DCA, and we allow all this context to be stable. Okay, when I run my simulation now, with all these structures, what you observe now on this plot to the right is that you get your open structure, you get your closed structure and do cluster analysis, but you get a third state that's a twist state. So what you see is this enzyme, as it's working, as it's binding the ribose, it's not enough just to close, it has to close and twist to the ribose to bind and then fully close. We can see that on this final slide. You see that it twists and then closes. And this tells you the power of DCA of predicting these things. So I could come to you and say, now I have a prediction power. This state is now being observed on detailed calculations and being proposed experimentally based on some spectroscopy data. But you say, am I predicting? I'm not predicting. Evolution is predicting. I'm just using my methods to extract this information from evolution and being able to predict that structure. So it tells you that based the sequence information can tell me about these functional states as I'm going looking into that situation. Now, how can I actually, as I told you, now use this information to go beyond the predict single structures, but actually be able to predict larger systems like complex of proteins. I don't want to do a single protein folding problem. I want to predict complex of multiple proteins come together. That's actually how we start the work. Actually, we start our work by doing that. Part of our group also work on this problem, what we call two-component systems. These are components that basically, where you have this histidine kinase and response regulator that basically gets phosphorylated from outside, and that's how information is transferring to bacteria. And you're trying to identify these pairs of contacts. And I'll come to this topic a little bit later, but let me show the initial results. The first system we did a prediction structure of that was the system based for SPO0B and the for SPO0B and the response regulator in Bacillus with Alex Shug, that was the group on that time. Let me tell a very interesting story about that. This was actually our first work with DCA and took us 6 months to publish. When we published, the crystal structure came back. We could actually be published 3 months earlier with a prediction on 3 angles resolution before the crystal structure was available. But we are insecure. So there's always the question in science, how fast you want to be to publish and how is low on to do. Actually, we're doing much better today. Basically, Ryan Shang and Farouk Marcus. Ryan Shang is still a postdoc in the group. Farouk Marcus, like I said, is a postdoc in Dallas, have been working on many of these pairs we're going to see before. The difference here now is that you have to create a sequence alignment, a multiple sequence alignment of not just single sequences, but you have to put the 2 pairs of proteins. So you create this sort of super protein. That's the protein in the bottom, the protein in the top, both come together. And you have to align the proper pair. You have to make sure the proper sequence that binds is here. In bacteria, that's very easy because the same pair of proteins have the same operon. So you always can identify which pairs have to come together. When you go to eukaryotes, it's more complicated. You're not going to have time to talk about it, but you can do by species. You can do similarity in species, but it's a more complicated. But anyway, you have to have this super sequence to do here. One case, actually, you can get a pair of proteins, but you don't have to do super sequences, is when you have homodimers. Homodimers are very common on function in all biology on prokaryotes and eukaryotes. Now, what we do on homodimers is, remember, you have the same protein. Now I look at my DCA contacts. I look which DCA contacts are on the monomer, and I remove them. And I just look, the contacts appear to be mistakes. They appear to be mistakes, but they're still contacts. And I assume that these contacts probably are contacts between the pairs, are the interface. They are created binding between the contacts. We can do further criteria, like make sure they are on the surface and stuff like that. But we can now start to identify which possible pairs I have into the protein. So going back to those contacts I predict, I assume the ones that are not contacts exist on the monomers alone, they're probably contacts between the dimers. And with these things, we can actually do predictions in better than 1.5 angstrom, 1.3 angstrom. That's a great work we did in 2015. Now it's used by many people. Here's a bunch of dimers that we do. What's very interesting, we can even predict structure of dimers that do domain swapping. They're based on the sync swap using the structural-based models for the system. Now you can see the precision is amazingly good that you get into these dimers as you come along. As you can see, things go always below 2 angstroms and can go even sometimes close to 1 angstrom. You get very good structural predictions of these dimers. Let's make life a little bit more interesting now. Actually, I told you about these proteins. Let's do a much larger protein. A much larger protein when I talk about is the structural methods chromosome proteins. The motors that keeps on chromosome makes loops. When chromosomes are formed together, these SMC proteins that I'm going to tell you a little about, basically condensing and cohesion that happens to the chromosome, they exist there. These proteins are gigantic. They're very, very large. That's work done by Dana Krupel, that's now in Israel and around in San Francisco. Both works on biotech, and with the group of Peter Wallens, with Nick Schaefer, and Peter Wallens is collaborating with us. So let me tell you a little bit about how this work was done. Like I said, these proteins, if you have been looking at chromosome structures, they are responsible for making these pairs. They are also responsible, like for condensing, is responsible to condense chromosome. They collapse the chromosome, like for example, when you go from the interphase to the mitotic state. Okay, now we are trying to predict that structure, but that's a gigantic structure. The way we do it is the following situation. Again, we have here, let me just go back one step. Actually, we have 2 big proteins that basically make this loop here. There are 2 SMC proteins, and you have the one that glycine. There are 3 or 4 proteins here in the bottom. So we make an alignment of this super protein with these 5 proteins. We do this multiple sequence alignment. And at the same time, we have crystallographic information for parts of these proteins. As you can see here, just so that you can have a look, you can observe they have 2 of these large proteins here, the base that is SMC proteins. You have 3 glycines here. And now in orange here, I'm showing are the little pieces of these 3 proteins that people have from X-ray structures. Okay, now what we do is we try to say, can we build the other contacts just by using now my DCA, by using covariance? And when I do that, now I get this black map that fulfill my entire system here. The first thing we can observe is that the contacts you're very interested are these contacts on these areas here. There are contacts between different groups. You can see, for example, here in our SMC as we build this up. Here's one SMC. Here's the second one. Here is the each one of these is a coil domain. You can see the first coil, and you can see here the interactions between the coils. You can see the interaction with these glycines. So the first thing we can observe, we can build a structural-based model, predict the full structure, and what we learned. Let's go to the next slide. First, look how big this protein here is. This protein is about 40 angstrom, okay? Here's the size of a nucleosome that you know that you have in DNA. That's about 10 nanometers. This is 4 nanometers. That's a very gigantic protein. The first thing is, by looking at this region, there was a big question if people said, if you had 1 loop or 2 loops. And what we know here is actually, in order to satisfy this context, we only have one SSPA and two SSPB proteins that goes in there, as you can see here, as I show on this contact map here. These are ones that satisfy these contacts. So from that, we can predict a structure and tell you that's the problem situation, and we have the right stoichiometry of this problem, okay? Now let's have a look at this hinge here for you to show you how this method is powerful. If you look this hinge area of this protein here, you observe the following situation. I'm just move this around, but this on my way. If you just look this hinge area of this protein, you observe the following situation. In origin is what people have from the crystal structures. And you can see here, by looking these contacts here, you can see that you have this green contact is exactly what you have on this closed structure. But you actually have these additional contacts here. So what we do here, not only we are able to agree with the X-ray structure just on the hinge area, but we see there are two structures in that region. That tells you that you have a closed structure and an open structure. So probably the hinge in the top there is opening and closed to let DNA to come in and let DNA to come out. And you can see from the electrostatic map that we have here, actually, on the open structure, you have the right thing to bind the DNA. Look, the DNA is negative, so that binds into this positive area you have here. So that shows you the enormous power of this model on doing that prediction, on letting you see the situation. Okay. So what we tell now that basically the power of these methods in order to predict these gigantic structures, and now we can think about it, how cohesion and condensing does that. So now is the full scheme we have these days now, use this coevolution method and structure-based model to get our initial structures, and then we improve them using better physical methods to predict the structures. And we have been able to predict the structures for these proteins. And now you can see how well they agree with X-ray structure, at least for the pieces that we know. The other piece we don't know, but we can use at least these pieces to confirm that at least part of the things which are very good, and we can predict the full structure. And with the full structure now, we can actually do calculations and show that these proteins actually exist on very many different structures with different, with different levels of twisting as they come around. And these different isomers of these proteins that are here can tell you that the question is which one they exist. And we have been looking with experimental data what people are trying to understand from cross-linking experiments. And we show to them, actually, the experiment can only be explained by using a combination of multiple states with different levels of twisting. And that's why they couldn't predict a single structure for it. Now, with this level of twisting, understand that these things can twist and untwist as they go around these proteins. We can understand how they interact with DNA. And the question now is one of the main points of research in our group now. Are they interact with DNA in a way they're just making loops where the DNA go across, like we show here, like handcuffs, or they actually DNA twists on it and you create what we call braiders. We believe this is the case, how these motors work, but that's an ongoing topic of research in the group right now. Okay, so let's continue a little bit into this subject. Now, what I told you before was that I believe that by using this DCA, by getting this probability function, I could predict contacts. But can I become a little bit more greedy? Can I say that these energies here that we get on this thing that we use for prediction are actually real energies? And by looking at these different AIs and IJs, I can see which pairs of contacts happen more often than which other pairs of contacts. And by doing that, I can see which pair of contacts are more stable than which more contacts. So I can get a real energy out of these things. So I can use evolutionary data to predict energies, okay? Here you have to be very, very careful because remember that basically you maybe start running out of data. For bacteria, we can do that. For our system, it's more complicated, but it's actually our challenge, this method, the limit these days. If that's true now, we can do mutation studies, computational mutational studies using the energy functions that come from the sequence aligned data that I have for these proteins. Here is an example for a few families of proteins where we compare delta G experimental with delta G from DCA. And you see the correlation is very good. Actually, although I'm plotting everything on the same plot, each of these families is computed separately, right? So I'm just doing mutations one family at the time, because doing all families together can become problematic, but that's one family at the time. The nice thing is these predictions are better than most methods to compute binding stability energies of proteins here. And doesn't mean that I'm a better calculator, just means the evolution has calculated. This stability is basic, as you can see, as the system evolves, there's a strong correlation between the landscape of sequences and the landscape of structures that I'm telling you about it. As you can see, delta G experimental is as units of energies. Delta G DCA, like I told you, is just DI is an information number. So I need something to convert. This thing has no units to a scale of energies. And what converts
You
is
seem to be
what
muted,
I'm showing
sir.
here. Here I'm getting a particular protein that's folded. Here I get now the same structure, but I run all the sequences, randomize them. It's what we call the molten glob. This is low, tell me what the stability is. This tell me T-select. T-select is a selection temperature that tells you as sequences are moving in evolution, they do mutations, they have accept new sequence. Just like the Monte Carlo way is which sequences that are a little bit more or less stable you accept is how far you go. So that tells you about the evolution landscape. So now we actually have a great system to understand how selection takes place and tells you, since you have this funnel landscape I told you about it, evolution is just looking for better native contexts as you go along, but sometimes you have to go a little bit higher in energy, and this select tells you how high you have to go. If you're used to Monte Carlo idea, it would be like a Monte Carlo in sequence space in real life for these proteins. So if I know how these proteins operate now, I can actually do functional studies, okay? So in order to do functional studies, like I told you, here's the case of proteins. And you see here, for example, that basic, like I said, that's a bacteria. And we have all these different histidine kinases. And the way the system works is basically the ones you want to send a signal, they get phosphorylated. The one that gets phosphorylated transfer from the kinesin down to response regulator. Goes into the kinesin goes gets the right response regulator. You phosphorylate the response regulator, and the response regulator comes all the way to your gene, and now you do gene expression. Now, the question is, how do you make sure you identify the right partners? These histidine kinases, they are very similar, and these response regulators are very similar. How do you identify just the right partner? Observe that in bacteria, you have on the order about 100 to 1,000 of these two component systems. How do you find your right partner? And what we did here is a very interesting story because, like, again, we start to look these interactions between these two component systems. We apply this energy function, and we try to see, can you find the right partner? So basically, we use that energy function. I told you about it, that I came back here. I'm sorry, I'm going back. I use this energy function I told you here in order to predict the context on this pair of residues. So what you get is here. Here are all possible pairs of response regulators together with their histidine kinases. If the systems were very, very precise, if they never made mistakes, you should have this diagonal very red and everything else shouldn't bind. What you see is our predictions are actually very, very good. Okay, you know that these things are not perfect binders because the way the system works in biology, you have many pairs. These things are a few of them, and you bind to one, and then you bind the other one, and you're just changing this equilibrium. That's how function takes place. Nice thing about it is basic. You can get a pair like, for example, this pair here that's very white, and that's because you have very little data. Or you can look, for example, RSTA and RSTB. These are very promiscuous response regulators. So you can see here that binds to lots of other things. So from this calculation, we show that actually this energy is very good, even when this protein is being very similar, to separate which are the real pairs of binders and which are the other pairs of binders. I want to get to a particular system here for you. One of these particular pairs, this is on Bacillus subtilis. I want to get this E. coli, I'm sorry. I want to get this 4Q, 4P pair here. That's a very interesting pair. You can see the signal is good here, but it's not great, but it's a good system to have a look. These systems actually work we did on molecular biology and evolution. This work, the experimental work here was done by MATLAB at MIT. What they did is basically this 4Q4P system is a system responsible for magnesium control inside of the bacteria. So you see, the kinesin 4Q get this magnesium, get this response regulator 4P that goes to the dimer here. What MATLAB observed is that the interface of binding has 4 residues here. So what they did is they mutated all these 4 residues by all possible pairs of 4 residues. So what they did is they had 1 to the 4 different configurations. Now, basically, they had 160,000 different variants, okay? We are into this variants idea with all this COVID these days, but see, they create all these things. And the question you're doing here is, when you do these mutations, this pair is still functional. And the interesting part is that about 1% of them continue to work. And what we come is say, is our Hamiltonian good enough to experiment here? This is not a quantitative measure in a sense, say which is a better binder or a worse binder. It's a piece of information. So when they did that, as you can see, we did a calculation into this system. I want to show you that I want to look basically. Our question is, can we figure out which are the better binders here? These would be the results we had in a very, very low energy. What we observe is, if we look our low energy states, the states are down here below the wild type that we expect to do better by our design method. We cannot do this high energy part here because something may be functional on better energy. But the question is, can we say there are low energy, the most stable binds are good ones? You can see we had about 80% prediction. However, what made this paper very interesting is the following thing. When I do a mutation, I can bind better to 4P, but mutation can also make you bind to the other different response regulators. So not only I want to remember, I want to go back here, not only I want to increase the specificity, a better binder for the one you want, but I want to bind worse the ones I'm competing against. So optimization to this protein design that base biology is doing here, and we can do, is not only you're trying to bind better to what you want to bind worse to the competition. So what we do here now is we see the ones we bind better, but also we remove the ones that we're binding better the competition. When you do both of these things, the design part to the state you want and undesign the competition, then I have 100% prediction. The method becomes very powerful. That tells a lot about why it's very hard to do protein design, okay? That tells you that basically the entire work is basically, you can state very easily, but when you're doing protein design and try to do things that work better in biology or trying to develop drugs, it's not just a question of binding better, it's also not binding better the competition, and you have to make sure that you don't have these things. The competition means that when you have side effect, drugs become becomes not effective anymore, and that's what makes biology and protein design a very complicated field to to work these days. On the last 5 minutes, I just want to tell you that these tools can be used for other problems, and we don't have too much time here, but I just want to show you that. Basically, one thing we are doing, a complete different scale now, is predicting structure of chromosomes. And the chromosomes have what we call different types that you call euchromatin and heterochromatin, in our case, A's and B's, and we try to learn this information from chip sequence information. The structure of your different chromosomes, for example, different cells that you have, they are different, but they're not different because they have different genomics. They're the same genomic sequence. It just comes from epigenetics. So I don't want to go in details because I'm out of time, but I want to show you one idea that basically, so what we have done here is being able, again, look different, basically different epigenetic experiment. That means I'm looking at different particular states, for example, methylation states, transcription factor states, they bind to different factors. These are different experiments. And now I look at covariance of those experiments. And by I look at covariance of those experiments now, I can predict these types A and B, like I told you. I want to go in details for you. But the idea that basically, when you're looking at data these days, I'm not just looking which particular, for example, particular methylation factor that can methylate particular point, a methylator, or does it, but I also look how different effects of epigenetics come together, and I can use these things to prediction of chromosomes. So it tells you this idea of covariance. It's very important because it tells you how things vary together, can tell you a lot about predictions as they come along. So I'm going to skip that because I want to stay on the time. I promise the people here, and I just want to thank everybody that has been participating on that. I think these people on the period of the last 25 years have been the people doing all these ideas on energy landscape ideas that come into this way. These are my collaborators. I want to thank all the people that have been financially supporting us. And with that, I want to stop here. I'll be glad to answer any questions you may have. Thank you.
Muchas gracias.
Thank you very much, Dr. José Onuchic, for the very interesting presentation, how from coevolution you can predict not only the structure of the protein, but also you can identify interactions between different proteins and, as we saw last, in the chromosome structure, which is totally different. Now, here I have a question from Camila, who asks if the forecast technique takes information of the environment like temperature or pH in part of the binding of the protein, or if this affects the predictions.
This is an outstanding question, okay? Outstanding question. The way all these techniques have been developed, okay? We are using the information from evolution. That means, Camila, that our results, in principle, they have been biased by whatever the environment on the planet did as the systems evolve, right? And so we haven't taken this into details, but I'm glad you gave me the opportunity of elaborating on that a little bit more. So the interesting question we have is that, for example, on this case here, we take all the information we have, not only whatever the environment was when these proteins evolved, but also all the species put together. I could, in principle, try to do DCA for different species at different stages, and I can tell you about some very interesting results. There, for example, cold shock proteins that bind at bacteria, but they don't bind at the human level. So if you look at their DCA, it's different at these different levels. Now, so that it's a problem because I don't have control over it. I just basically, since my laboratory is the laboratory of evolution, I'm stuck to whatever the data that exists. I look all the possible sequences we know about that particular protein, and they have been evolved on many different conditions. Now, that said, very smart people have gone beyond that. And I haven't gone, I'm telling you why. A lot of experiments I'm going to tell you about are done by Farouk Marcos that I told you about. That's former postdoc of mine. That's now professor, University of Texas. And he just got promoted to associate professor. But my policy is always when my people come out of my group and they're assistant professor, I don't compete with them. So he was doing those experiments. And what he was doing is what we call laboratory evolution. So with bacteria, you can actually create experiments where you put these colonies of bacteria, and now you change conditions into the laboratory, and you can start to see effects like that. And it's very nice to see how, you can read some papers he did on that, that basically you can start to see, for example, different evolutions and different binding partners, because, for example, the bacteria are under different stresses. For example, in the case of response regulators, you want to bind to different things to respond to those stress. So you can see, depending on how we evolve the systems on different conditions, they may evolve to different systems, and it's a very interesting question. So we don't take this into account. These studies are in the beginning now. In our case, we're just using the entire landscape of evolution, and you can, and then and we are bound to it. All the good and the bad comes with it. But new experiment, what you call this sort of direct evolution on the laboratory now, people are starting to do that base. You can create your own DCA system, right? By putting these bacteria on different stresses, get different sequence and see what happens to your system.
Muchas gracias por la respuesta. Yo tengo una.
Thank you very much for the answers. I have a question, Professor. It's a different what you've done of using the covariance or the correlation between the coevolution of the amino acids for interactions between themselves. We have seen that these mutations or coevolution can also be compensatory mutations. I want to ask again, I got tangled, but if these mutations, these commutations can also be indicating certain compensatory mutations related not with proteins, but other binders, maybe a substrate or even interactions with RNA or DNA.
Oh, your question is absolutely fantastic, okay? So what you are seeing from your question now is that these ideas can evolve enormously, right? When we start these coevolution ideas that you did today, and I didn't want to complicate more than I should, I was already complicated enough. I just put the possibilities that basically you have these things coming together. But if you think about statistical possibilities, you have to add all the other possibilities that come into the game. One of the situations that we are discussing now are, for example, as we get to intrinsically disordered proteins, for example, I'm going to go to DNA. But let's start just in the protein case. That's a little bit simpler first, okay? And you observe that, for example, different IDPs may bind to different proteins. Okay, and actually, depending on the partner they have a different structure. So as I look these different dimers, these proteins with different structure, can I now do evolution and make sure how the sequence information actually now adds the information, not how to bind to a single partner, but binds to a second partner. So what we're doing now are studies when I try to look the coevolution of pairs of proteins, but also trimers of proteins, putting all these possibilities together to come together and see how evolution has selected for that. Right? So your point comes to that regard. Also, what you're trying to say is, what's a very good point, is that is that, for example, if DNA is present, RNA is present, they may themselves affect the structures. That, in general, is not only a nightmare for us, how to take all that into account, but that has been the nightmare on the entire structure prediction game of proteins. I can have AlphaFold, I can have the most complicated proteins, and someone tells me, can you predict the structure of this protein? But they never tell me there was a DNA around. I may get the wrong structure, right? So basically, so one of the things that's right now, they're coming together. Now, the beautiful part of it, you can always get a negative and make a positive, right? Basically, people used to say this in life. If life gives you a lemon, make a lemonade. In Brazil, we have a change. We say if life gives you a lemon, make a caipirinha. Anyway, you can make different versions of it. But the idea is, if we start to predict wrong structures, when you believe your methods are sufficiently good, and you start to see different experimental information, that may be a hint for you that the process is much more complex and something else is involved. So in a sense, you have to take both ways. On the old days, people just want to say, oh, I want to be able to do structural prediction. Now that our methods are becoming very good, you can actually say, if our predictions don't agree with the experiment, something else is going on. So that means there's new science, new mechanisms, there's a third partner that's involved here. And DNA and RNA are particularly complicated because they have charts. As I told you, basically, I told you a little bit about the work we're doing with these SMC proteins. And now we're trying to put the DNA into the game and try to see how this model works and really complicate the game. But your point is very well taken. And I think that's something that people are going to move into that direction. It's very interesting how to put all this information together.
Todo complicado. Consuelo pregunta.
Consuelo asks, how long do you take to make the DCA calculations, and how much computer resources are required to make the predictions?
DCA calculations actually are very inexpensive, okay? The most complicated part into this game is actually doing the structural-based models afterwards and running the simulations, okay? I can tell you that our website for DCA, dca.rice.edu, actually, you can actually put your sequence there and get the information, run DCA if you have your MSA for free. It's cheap enough that you can run our server. The structural-based model simulation a little bit more expensive. We have the sites called smog2.org, that's basically that runs all the simulations, and you can lower the software to do that. Now I can tell you the good things about all the simulations. If you look at our website, particularly SMOG website, we have everything convert into what people call OpenMM. OpenMM is open source software, and you can run on GPUs. So now basically many of this system, my students can run on their own personal computer because these GPUs are getting awfully fast for simulations. I can tell you that our chromosome simulations that you're running on normal CPUs, now we are almost running them 1,000 to 10,000 times faster with GPUs. And if you have multiple GPUs, even better. But basically, simulations are getting cheaper as they come along. But if you want to talk about how to do simulations in details, basically, feel free to contact me and I put you in contact with the right person that can help you. Okay?
Y la última pregunta de Camila también, que pregunta.
And the last question from Camila, who asks a little bit about what we talked about RNA and DNA, but if there's certain complications for the forecasting of eukaryote proteins simulations, if the RNA sequence suffers modifications, transfictional modifications, or those are already taken into consideration as before, or after everything.
Like I said, when we do the proteins, okay, So let me tell you a couple things that happens when you do protein-protein interaction, okay? When you do protein-protein interaction, if you get a bacteria or a system like that, you want to make sure which is the real partner between those proteins. Like I said, in bacteria it's easy because they share the same operon, so it's very easy to identify. At least they share on 98% of the time, so you make very few mistakes. In humans, if I get a particular protein, if I'm studying a HSP70, whatever, okay? Then I have to know HSP70 bind to HSP90, which are the pairs. I can divide by species, but sometimes you have multiple pairs on the same species, and how do you know which ones go with which ones? That's a very complicated story. Martin Waite does this by some similarity analysis. They're very complicated papers to do that. When you talk about RNA, it's becoming a very interesting game. Let me step back from your question a little bit. Alex Chug, that's the one you saw, I told you about, did the first DCA work in 2009 in our group. Got very interesting in trying to predict structures of RNA, and he created the DCA for RNA now. You can find the DCA for RNA. The problem now of doing DCA for RNA is that basically proteins have 20 amino acids. RNA has only different 4 bases, right? So basically, it's very hard, there's not enough discrimination. So Alex Chug is working by doing now, instead of looking at sequence every base pair, he do pairs of base pairs and try to do all this alignment to multiply its effect. They're getting some interesting results on that. For Marcus is interesting these days about binding of proteins with DNA and different RNAs. And then what they do is direct evolution and look the data, and then they look the binding assays at the same time. So in that case, you can do now which are these structures, what's this model, and which of these particular proteins are bind to bind RNA are not. So these are very interesting questions you're asking. And I think that's what people are doing these days. On the case of binding, he has to do both binding experiments together with sequence alignment together, and then he can extract all that information. As you well know, basically, that becomes very interesting because you may have things that have the same structure, but have a different charge distribution on the surface, right? And that may affect the binding. So when you look this complex, I think where you're coming from is, can we just do sequence analysis to do that? And I think you have to go beyond. The DCA approach on itself just look interactions between proteins, right? And we are averaging everything else, right? When I get the entire evolution, I'm averaging everything else. Now, that may work in some cases and may be very stupid in other cases, okay? For most of the proteins we do, like you can see from some recent work from Farouk Marcos, and I recommend you to get in touch with him if you want to use Dallas, then he's trying to do, together with that, binding experiments, and the combination of both can start to bring information to that regard.
Muchas gracias. Y bueno, ahora sí, la última pregunta.
And yeah, the last question now. How do you forecast the binding of the conventional analysis as based, for example, on AI? The conventional structure analysis with a new tool based on AI as a combination of structures and the new AI technologies.
Okay. As you can see, in a certain way, let me tell you the following thing, okay? I love AI because I have been on this field more than people like to do that base. I was a graduate student at Caltech from 1982 to 1986. That gives up how old I am, okay? And at that time, I was working with John Hopfield when he was creating the first papers on neural networks. So you're at Caltech when the first neural networks paper came together, you're trying to figure out how can we put this data into this particular basis. If you think about it, the inverse problem, when I told you, you get all this covariance matrix and you try to put into an energy function, that's an AI problem in a sense, okay? Basically, you're trying to learn from the covariance data, you're trying to learn the energy function on this case. When I do my prediction of energy, that's AI. Now, you can do different levels of AI depending on what data we have. But our experience in biology is that supervised learning is the one that works the better. One of the problems you have with AI, the question is, you run into trouble when you don't have enough data, and you run into trouble when you have too much data. And I'll be glad one day, if people want, I have my theoretical biological physics class, and I'll be glad to go over my series of AI lectures with you guys at some point, but tells you that basically, if you put too much data, you overlearn. And that means, and when you overlearn, you start to fit noise. So the system gets worse. So the question is now how we do that. So when you do the supervised learning with one model, you know more or less what the complexity of the system that you have. Okay, and you know how many parameters you have. So you can evolve into different levels of data and start to look at the Hamiltonian you get it to the end. In our case, it's like the one we have for prediction, and start to see, this is Hamiltonian change. Is that getting better? How the predictions go? You can apply to trivial cases and see how it does. So I think the idea of doing modeling together with AI is the real thing these days. Otherwise, it becomes very complicated for you to know which level of neural network do you want as you couple your systems. And that's one of the things we are having. But you see, on the idea of the chromosome data, I didn't go into all details. We have lots of these physical models where we use AI from the experimental data to learn the parameters of the model. So it's a mixture between doing modeling and data analysis. And I think that's the real success for the future.
Muchas gracias, profesor.
Thank you so much, Professor. And aside from that, okay, we'd like to especially thank you for giving us this great lecture. And also, we'd like to thank the people who were here with us, who they really liked everything, and we'd like to thank them for your question. And at 11:30, okay, we are going to continue with the Forum for Women in Science. Okay, don't miss it. So thank you so much.
Thank you very much.
Fue un poquito. Todavía no se vaya.